Sure. I'll start off by introducing myself. So I'm Jennifer Carbrey. I'm currently a faculty member at Duke, but I guess started my scientific training at Clemson University, which is a public school in South Carolina. I got my bachelor's degree in biochemistry, and then I was able to basically have my graduate training paid for by going to Johns Hopkins University School of Medicine to get my PhD in biochemistry. And so I chose my graduate mentor, Dr. Peter Augrey, and in his lab I studied yeast aquaporins and also Aquaporin 9, which is found in, for instance, the liver in mammals. And so I finished up my graduate work, and then I also continued on in Peter's lab. And so I was lucky enough to be there for many years before he won the Nobel Prize, and then I was there when he won the Nobel Prize, which was a really amazing experience to be a small part of. So I heard that he had won the Nobel Prize in the morning before I was heading to lab on NPR, actually. And so I headed in, and we all gathered and waited for Peter to arrive, and we lined the hallway once he arrived and gave him huge applause. And then Johns Hopkins put together a press conference, and so we got to go to that. We had celebratory dinners and receptions. Another interesting thing was that the BBC came to our lab and interviewed us and interviewed Peter, so that was really interesting. And then we also, some of us from the lab went to Stockholm for the time when he was actually receiving the award, because, for instance, the Nobel lecture is open to the public. So we were all able to go and see him talk about our research and put our pictures up as he spoke and gave his Nobel lecture. So it was a really incredible experience to be lucky enough to be a part of the lab at that time. And so then Peter moved here to Duke, and I came along and became a faculty member to run his lab. And then he went back to Johns Hopkins, and that's when I found a position here on my own where I teach full-time. So I teach mostly medical students, some graduate students, and then a colleague and I also have a Coursera course. So that's free to anyone in the world. That's an introductory human physiology course. So I give a plug for having a PhD. It's really nice to have a degree that's really versatile. So I was in research and found that I really like interacting with people a lot, and I really love teaching. And so I'm grateful that my PhD allows me to be versatile and choose a wider variety of careers. So any questions? I guess we can move to questions.
Yeah. We'll begin with our rotation then, where it'll be Everest, and then I think Steamboat Springs, Waukesha, and then the virtual school. We'll have to download the emails from the Colorado school, so we'll see if we can get that to happen. So I think our students are ready here for some questions.
Hi, our first question was on the function of aquaporins, and we were wondering, how would you describe the rotation of the water molecule and the functioning of the NPA site?
Yeah, I did not study the structure very specifically. I wasn't involved in the crystallography work, so you students probably know more about the structure than I do, actually. So I just know that the channel is, some important aspects of the channel are obviously the diameter, that being restrictive to certain molecules, and the positive charges repelling other positive charges. And then some of the hydrophilic spots in the pore that help to move the water quickly through the pore. And then also the hydrogen bonding that isn't too strong, but strong enough to remove the water from the other waters around it. So that's about the extent of what I know about the channel on the very molecular detail.
All right, our second question is on aquaporin gating. So how do aquaporins gate?
Right, so most of them actually don't gate. There's only one that does is Aquaporin 6, which is permeable to anions and actually is gated and opened by mercury, but it's also thought to be opened by acidic pH. But most of them are present in the membrane constitutively and aren't gated. And if you think about it, for many of them, that is fine. So the fact that they're always open and able to move water is fine, because in our bodies, usually the osmolarity is going to be equal on both sides of the membrane, and there won't be dramatic changes where having an aquaporin open would be a problem. But then when it allows water to move when it does need to be, when it does need to happen. Okay?
All right. And then another question that we have is, well, we're trying to focus also on telling the story and the repercussions of aquaporins maybe malfunctioning and some of the applications for aquaporins. So what applications do you foresee as being the most promising for aquaporins?
There are clearly some that are involved in diseases, and so that's obviously important to understand. And if we could find really specific but not toxic inhibitors or activators of aquaporins, that would be certainly useful in certain physiological circumstances. And then there's just the idea that understanding completely how the body functions is also important. So there's still a lot to be learned about how aquaporins function in certain situations or in certain tissues. And even if losing an aquaporin isn't fatal or doesn't cause a noticeable disease, it's still important to understand how they function.
Sure. Okay.
There's our three questions. I don't think we have the Virginia school here, so is the audio fixed at Colorado? Cindy, is it possible?
Is there any potential for using CRISPR systems to repair aquaporin mutations?
Yes, that would certainly be a possibility, just because CRISPRs are such a powerful technology. I think that for the most part, the aquaporin diseases aren't so traumatic, and the technology for how to deliver the CRISPR isn't well established enough for them to be used. But no, that's a good idea, particularly for the nephrogenic diabetes insipidus, where there are aquaporin 2 mutations, where those patients have a really debilitating disease, and so the risk of using CRISPR and whatever side effects that might cause would be worthwhile. So in the nephrogenic diabetes insipidus patients who have mutations in aquaporin 2, then that means that they can't concentrate their urine, and they're producing something like 15 to 20 liters of urine, which means that they're having to drink more than that every day. And so, yeah, it's definitely an exciting technology, and it'll be interesting to see how it gets used in the future.
How do the structure and function of aquaporins in humans differ from those found in plants?
Oh, the structure is very similar, but the function is going to be quite different, and I am not so well versed in the plant aquaporins anymore, but obviously they're dealing with trying to get water in from the environment, which is completely different from one of our story wells. So most of our aquaporins are kind of dealing with water within our bodies, whereas, for instance, the aquaporins in the roots of plants are trying to get water from the environment. So yeah, I am not so well versed in the plant aquaporins to be able to talk about that with much detail.
Third question.
Are there molecules besides mercury that inhibit aquaporins, and are there instances in which the body needs to inhibit aquaporins?
So there are some other small molecules that affect some of the other aquaporins, but mercury is definitely the most most versatile of the inhibitors, and obviously it's toxic. And really the main way that the body would inhibit the aquaporin is by controlling its localization. And so aquaporin 2 would be our main example of that, where it's kept in vesicles until vasopressin is released from the pituitary. So if you've been jogging outside, you've lost a lot of water, and so now you need to make more concentrated urine, then the pituitary will sense that increase in osmolarity in the blood, cause vasopressin to be released, and that normally causes then aquaporin 2 to come to the membrane of the cells in the certain part of the kidney in the collecting duct and allow water to leave the forming urine and concentrate it. So that's really the best example of control, and it's not really an inhibition, but it's at least controlling the localization of aquaporin 2. Then there are other aquaporins that are whose function are controlled by things like pH as well, that can affect water transport and not affect localization, but those effects are somewhat subtle.
Great, thank you so much.
Oh, you're welcome.
So the three from Colorado, can we move to Virginia, to Hanover?
Hi, we don't have any video, sorry, but we have microphone access, and we just got on, so we're sorry we're a little late, got a little lost in the crowd. So I'm not sure what you've already covered, and I apologize if this is a repeat question, but how was it? Like, how did it feel to be part of that team?
Yeah, it was really fun. It's one of those things even though it's been so many years, it's been more than 10 years, I still can't believe it really happened to me. But then I had such a small part in it. So it was more just an amazing experience, just to see what the process is like, and to, yeah, it opened up many opportunities to just meet different people from around the world. We had people calling the lab who had medical issues, hoping that they could be helped. I mentioned earlier, we had the BBC in our lab videotaping and interviewing us. So it was an extremely amazing experience. And then a lot of us from the lab went to Stockholm to see his lecture and to go to a couple of other events that were happening when he was receiving the award. So it's really fun, and it's fun to sometimes let slip to my students that I was in a Nobel Prize winning lab.
That's awesome. Yeah. I think that's it for us right now.
Okay.
Yeah.
If we could have Waukesha next, please. Mr. Osmond and gang.
Okay, so I see the 3 questions. What information about previous cell models caused you to suspect something was transporting water other than directly through the cell membrane? Yeah, so those were actually studies that were long before the involvement of Peter Augrey's lab. There are observations of frog skin that was permeable to water and observations of red blood cells being permeable, as well as kidney tubules. And so, as well as the observation that mercury inhibited that process. And so it's one of those things now with molecular techniques and knowing genomes, it's hard to imagine how difficult it was to try to identify the protein that was causing the water transport when it's right there in front of you and you're seeing the water move, but yet you can't figure out what the protein is that's mediating that. And so there were other models that were used to study water channels. It's just the molecular identity wasn't known. So that was question 1. I didn't get the chance to read question 2. I don't know if you can flash it back up there again. Oh, when you were looking for a protein when you discovered aquaporins or different, sorry, were you looking? Yes, so the Augrey lab, and I was not in the lab at the time, but they were trying to study the RH protein that is present in people but not in others and can cause complications with pregnancy. And so so I'll check the messages button here. They were using gels to look for the RH protein, and that's when they found this other protein and that was a similar size. So red blood cells are pretty simple cells, and so you can run a gel of the proteins in them and identify many of them, and so that's how they found this other band that they thought was related to the RH protein but wasn't. And so they got an antibody to it, and that allowed them to study it further. So that's the protein that was distracting them and caused them to find the aquaporin. And then the third question is, did you face any struggles as a female scientist? So that's a really interesting question. There are some struggles. I think they're not nearly the struggles that scientists, for instance, my parents' age, who are in their 60s and 70s, faced. But there's just sometimes subtle things that I think people make assumptions. So, for instance, I have kids. I'm one of the few people that I interact with who's a female and young and has kids. And so, you know, I've had situations where people have kind of assumed, oh, well, she has kids. She won't want to be on this committee that meets at 5 p.m. in the evening. And so, you know, those sorts of assumptions aren't helpful. I can make my own decisions. You can ask me to be on the committee. So just, I'd say somewhat subtle things like that. But that's not much compared to, I feel, what people before me had to deal with. But during my training and certainly my mentor, I did not feel that it was much of a disadvantage at all. Yeah, and especially in graduate school and certainly medical school, there are just as many females as males. Okay. So I think those are the 3 questions from that group.
We could go to Wisconsin. Karen?
Okay, thank you. I think you've answered a couple of the questions that we had. But the other thing that my team was really interested in was what did it look like once you had discovered Aquaporin? How did you professionally reach out to the scientific community? What does that look like? How are you communicating and building on that knowledge and sharing that knowledge, and kind of how does that, what does that look like?
At the time when it was discovered, it had been sought after for so long, and so many people were trying to discover it that it was almost a problem. So basically, Dr. Augrey and his team, I wasn't there yet, had figured out what the protein was that was the water channel. And then they had to kind of keep it to themselves as best they could before they could get it out to publish it. So because once you released some of the amino acid sequence, then it would be infinitely easier for people to then clone it themselves and then try to beat them to be the discoverer, which is going to be the first person to publish the observation. And so that was actually, I think, an extremely stressful time, is once they figured it out, but yet they're trying to put together the right experiments to make a really strong argument and a good paper to show that they did find the water channel. And then after that, it was obviously a prominent paper, and the publicity and the announcement was just done because of the publication. And so then many other labs started studying aquaporins pretty quickly. So hopefully that answered your question.
It does. Thank you.
Okay, then it is okay, Dr. Carbrey. We'd like to begin a second round.
That sounds good.
So our fourth question is, why might aquaporins be suitable for drug targets?
Why would they? Is that what you said? Sorry.
Yeah, why would they be suitable for drug targets?
Oh, they would be suitable in different situations. So, for instance, if someone had an aquaporin 2 mutation that couldn't traffic to the surface, and so they have nephrogenic diabetes insipidus, if you could somehow bind a small molecule that would allow it to traffic, that might be helpful. Or there are examples of conditions involving injuries to the brain where having aquaporin is actually can be somewhat detrimental, and so it'd be good to be able to inhibit it with a small molecule or a drug. Or there are times when it's beneficial, and maybe you'd want to be able to upregulate expression. So yeah, there are lots of situations, and probably many more that we don't understand yet, when it would be helpful to be able to modulate the activity of aquaporins.
Okay, thank you.
Why do red blood cells contain a lot of aquaporins, and how do aquaporins contribute to the functioning of red blood cells?
Right, so that's a great question. I saw that one ahead of time, and I really like that one. Because it's ironic, because that's where they were discovered, and it's really not so clear. So they have a fair amount of aquaporin, and our blood is kept by the body at 300 milliosmolar, so that the aquaporin really doesn't have a gradient across which to move the water. The best guess is that there are times when the blood is going through, especially in the kidney, places where the osmolarity is different from the normal osmolarity. So part of the role of the kidney is to concentrate urine, and so to do that, it increases the osmolarity in part of the kidney to be able to concentrate the urine so that the water can leave the urine and it can become more concentrated than the osmolarity of our blood. And so one idea is that to be able to cope with that, then the aquaporins let the red blood cells swell and shrink, or shrink a little bit, in order to not just completely lyse. So that's just a hypothesis, but there's not been a phenotype found in terms of red blood cells in AQP1 or Aquaporin 1 null mice or humans. So it's not clear exactly what the role is in red blood cells.
Okay. Are there some personal experiences that relate to aquaporin research that you would be able to share that help put a face to why aquaporin research is so important?
Yeah, really, obviously it's a very basic, the reason why it's gotten the attention that it's gotten is because it was a very basic mechanism that was somewhat slow to be uncovered, basically. And so then the finding that they are present in all sorts of organisms, even yeast and bacteria and things like that, just show its importance. And then the other reason why aquaporins have gained prominence and why the Nobel Prize was awarded is also just its role in being one of the first membrane proteins to have its structure really well understood. And part of that, the reason why it's been able to be accomplished with aquaporins is just that it is a really simple and compact membrane protein. There aren't lots of loops that are hanging out. There's not lots of extracellular or intracellular domains. It's just very compact and in the membrane. And so the fact that it's one of the first membrane proteins to be really well understood structurally also makes them important.
On to Hanover then.
Hi again. We have some questions. What were your favorite or most enjoyable parts of the process as far as the research goes?
Yeah, it's obviously getting good results. And that often comes infrequently. It's a lot of work. You have to really love research to be able to do it. But when you get a good result, it's just amazing. And then the other struggle with research is often just kind of getting the premise for a publication. So you can get some small results, but once you finally figure out, okay, this is going to be the main point of the paper, I've got that established, and now I just need to add more experiments to prove my point or to figure out more details. That's always a good place to be in, is once you're ready, you know what your paper is going to be about. And then also it's just, I think research is fun just in the interactions with people. So our lab was also always a very international lab. We always had people from all around the world. And so getting to interact with people from all sorts of different countries and find out really what their lives were like and their views on the world is really, really fun. I still have many friends who are now spread out all over the world who I can visit.
That's awesome. Are you, and I'm sorry if we missed this since we were late, are you currently doing any research?
So the little bit of research that I'm doing is in education. So I teach medical students mostly, and I also teach graduate students, and then a colleague and I have a Coursera course. And so I'm exclusively teaching.
Okay. Is that difference between doing more hands-on research to education? How does that feel?
Right. So it was a conscious choice. I love teaching and I love interacting with students. And for me, that's what I need. So for all of these students who are watching, really, that's one thing to consider when you're thinking about a career is what you want to get out of it. And so I want to feel like I'm really helping people, and research is certainly helping people, but it's somewhat slow and it's not direct. And so with teaching, I can have an interaction with someone and they can say, oh yes, now I understand it. And to me, that is what's fulfilling. And kind of the great thing about having a PhD or an MD is that you can kind of make those decisions, because I didn't have that figured out when I was in high school or going to college. It was once I tried research and realized how much I loved teaching, and tried teaching and realized how much I loved that I was able to kind of steer my career in that direction.
Awesome. We have one more question.
It might be, you might not be able to answer it, but while you were researching aquaporins, did you ever find like a certain type of aquaporin that was unique in different specimens and animals, or were they all kind of the same in a way?
So a new different kind of aquaporin, is that what you mean?
Yeah.
Yeah, so is that, yeah?
Yeah.
Okay. Actually, my PhD project, when I joined the lab, it was 1996. And the yeast Saccharomyces cerevisiae, its genome had just been sequenced. And so I worked with a postdoc in the lab, and we were able to just search the genome and find 2 aquaporins, and then do studies about those. So I got to do it the easy way because the genome had been sequenced. It was the first eukaryotic genome that had been released, and so we just got to search and then put those RNAs into frog eggs, and so to cause those xenopus frog eggs to express the aquaporin. And then when we put them in a dilute solution, they swelled and they exploded, and so we got to see that they really were a functional aquaporin. So yeah, I got the fun of helping to discover an aquaporin, but with the genome being available, it made it much, much easier than it was previously. And that's another interesting thing, was just the huge hurdle of just finding the first aquaporin. Then once you have that protein sequence and you have some amino acid sequence that hopefully is pretty well conserved, then it's much, much easier to find the others. But until you get that first sequence and know some of the amino acids involved, and without a genome, then that makes it very difficult.
Okay. Thank you.
You're welcome.
I think that's it for us this time.
Try again. Do it one more time.
How do protein-on-protein interactions affect the function of aquaporins?
Oh, that's a good question. Some of that is not well understood. One example that comes to mind is Aquaporin 4 that is found in some cells of the brain, and bound by proteins and localized into these sorts of crystals because they're highly concentrated in certain parts of the membrane. And that interaction and why they form those arrays is not exactly clear, but the main protein-protein interaction would just be with Aquaporin 2 and getting it to the membrane. So that's going to be protein mediated. So Aquaporin 2 is going to be phosphorylated, and then it's going to interact with proteins that let it get to traffic to the membrane and then be in the cell membrane. Other than that, I'd say that that's not well understood, how many other protein interactions there are with aquaporins, and if there's a lot of regulation through that sort of route.
Could you describe some of your experiences with studying the structure of aquaporin and how the X-ray crystallography process helped you study the protein?
So I was not involved directly in those experiments, and actually the way that it worked for the Agri lab is that we would, our kind of expertise would be in purifying the protein and then giving it to the crystallographers, who are the experts in making the crystals, and zapping them with the X-rays and analyzing the data. But I can say that purifying them is also challenging. So it often requires growing liters and liters of yeast and bacteria, and then finding a way to lyse them and spinning down the membranes and using detergent to break up the membranes, and then using some sort of column to purify. So it's a messy process, and it takes many, many days. And then the other thing we would do is then be able to put the protein that we purified and put that back into membranes that we would make, and then we could watch the swelling or shrinking of those little vesicles, those little membranous spheres that we made, and be able to study the water channel properties of the purified protein. So those are the sorts of experiments that I was involved in, as well as the experiments watching the frog eggs shrink and swell. So that's how I learned how to do surgery and suture.
What advice would you give to a high school student interested in studying biology?
Yeah. So I would give you the advice of studying really hard, working really hard, obviously, and then just taking every opportunity that you can find. Obviously, you don't have to figure out if you want to pursue a graduate degree or not while you're in high school, but to know a field of study that you're interested in high school is a great first step, because then you can take lots of science classes in high school and then pick a major in college that you're really interested in. So basically, making sure that you love the topic that you pursue is important, because if you're not going to be able to make yourself work hard otherwise. But then thinking about really what you want to get out of a career and what you want your career to be is really useful. So with biology, there's just so many options of basic science versus medicine, and there's so many different types of practitioners of medicine that there's really a lot to explore. But luckily, being in high school, you have lots of time. But that idea that I mentioned before of how you want to interact with people, how you want to help them, how important it is that you feel like you're helping them, figuring those sorts of things out is really helpful. And I know it's hard to do it when you're young, but the other thing is not to stress about it, I think, too much, because with science, it's so flexible. So even if you're like me and you realize that there's other things you want to pursue, it's not that hard to do. And the other thing I'd give for science and careers in medicine is just that it can be really challenging, which is great, and it's also a great way to travel. So I've been able to go to conferences all over the world. So it's and have my education paid for. So it's a great field to pursue if it really interests you.
Could there be a way to make some kind of water filter out of aquaporins, somewhat like an artificial membrane full of aquaporins, so that only pure water would get through?
Right. So that I've seen some of the publicity or some of the information about using aquaporins to filter water, And I'm not sure exactly how that works, because when aquaporins work, they're moving water that's more concentrated on one side of the membrane to where it's less concentrated. And so it's hard to imagine how you could purify the water unless you had something on the other side of the membrane that would draw it, and then an easy way to remove that substance that drew the water, if that makes sense. So water, when it crosses aquaporins, is always going to go down its concentration gradient, which means it's going to move away from the pure water side and go to the salty side. And so I don't know if they use pressure or something like that to move the water kind of against its gradient and against the way it wants to move through the aquaporins. So I'm not clear exactly how that works. It's kind of anti-intuitive to me.
Waukesha, then, I guess. You had some questions on your message board?
Oh, yes, I see a question. How and when did you know that you wanted to be a scientist? So I feel like I knew pretty early, and it was in high school. My high school focused a lot on chemistry, and I really enjoyed biology as well. And so in high school, I knew that I wanted to pursue a degree in science in college, and that I was pretty sure I was headed to graduate school, and I knew that in high school. So I was exposed to a lot of science in high school, and that helped. And then I did a little bit of research in a summer program when I was in high school, and that was helpful. And I also shadowed a physician a little bit in high school. So those were some of the experiences that had me thinking that way in high school. And then once I got to college, I did research for a year and did a senior thesis. And so I enjoyed research then as well. And so that's why I applied to graduate school.
The only thing I wanted to add as far as a question was, and you might have covered this a little bit already, as far as therapies and moving forward as far as applications and the areas in science that are using this aquaporin research. Could you talk a little bit more about that?
Yeah, so it's, I guess there's many levels to that. So one is that a lot of our observations are in mice. And so that means that before we can really draw conclusions about what is happening in humans, then we have to study humans. And then the other issue with that is just doing studies of different situations in humans. So losing an aquaporin isn't often, it's not very, losing an aquaporin often doesn't lead to a really disastrous phenotype. So when we knock out aquaporins in mice, very often we find only subtle differences in the way something functions in the mouse. And even then, it's when it's stressed under certain circumstances. And so it makes it hard to know then and do the studies in humans when it's something often so subtle. There are some really obvious ones, examples of disease, and that's that nephrogenic diabetes insipidus that I mentioned, which thankfully is fairly rare. And then some mutations in aquaporin 0, which is in the lens of the eye, that can lead to really early cataracts. But so often the effects of aquaporin disease are pretty subtle, and so that makes it hard to do studies in humans and hard to come up with therapies as well. So it's not like some proteins where it's a very devastating disease when you have a disruption of the protein. And then the other thing is so many of the organs and tissues that really need aquaporins have many different family members. So that's true in the kidney, for example, so that there's also some compensation. And so that also means that often disrupting an aquaporin doesn't necessarily cause a very serious disease.
Thank you.
Mm-hmm.
Did we see another message from Waukesha by chance?
I don't see one.
Yeah. Do you have time for another round of questions? Or
sure.
Questions?
Yeah.
Susan?
What were original theories regarding the transport of water prior to the discovery and research of aquaporins?
Yeah, so it was clear that there were some tissues that were really permeable and some that weren't, and that because of mercury, that there was obviously something that mercury is binding to that was inhibiting the water transport. And then there were also studies looking at how rapid the water movement was in those tissues, and that suggested that it was protein mediated. So that's another important part, something to mention, is that water transports and moves across the lipid bilayer somewhat quickly. It's just aquaporins obviously make that much more efficient process, and so they could look at the amount of energy that it took for the water to cross the membrane and realize that it was protein mediated. It was just, the question was which protein was doing it.
Okay, what changes in technology have enhanced exploration of proteins such as aquaporins, and how has research changed since you became involved?
Right, so the main thing is just the massive amounts of DNA sequencing that are occurring, and with the availability of genomes, it makes it so much easier so that you can just, in literally just a few minutes, find candidate genes and then quickly put them into an expression system to see if they transport or what they transport. And so also the use of knockout mice, so mice that have the aquaporin, the specific aquaporin deleted, so that you can see the effect of not having that aquaporin. That's another tool that has really changed the ability to look at the effects and the role of the aquaporins in certain tissues. So that's another technology. And since I joined research, I haven't been in it that long for things to have changed that much, I would say. I entered just when all of those technologies were really getting going.
Okay, and then our question is, how and where are aquaporins distributed, and can they be transported or released as needed?
Sorry, I kind of missed that a little bit. Could you repeat it?
Yeah. How and where are aquaporins distributed, and can they be transported or released as needed?
Okay. So distributed in terms of organs in the body, they're in many different organs of the body. And then the transport, really the main example is the aquaporin 2 in the kidney cells that move to the membrane when you're thirsty and when you need to concentrate your urine. And then there's another aquaporin, Aquaporin 6, which is also in the kidney, and it also seems to be residing in vesicles and then move to the membrane. The role of Aquaporin 6, though, is not clear. It transports mostly anions, so ions that are negatively charged. So that one's pretty fairly mysterious. So that's the main control in terms of localization, and in terms of where they're distributed throughout the body, it's many, many organs like blood vessels, red blood cells, liver, fat, lung, kidney, certain cells of the brain, actually several cell types in the brain, so they're all over the place.
Thank you.
Mm-hmm.
Does Virginia have any more questions?
No, we're good. We're just listening and enjoying.
Steamboat?
I think that we are fine. Thank you so much.
You're welcome.
Can you see that message, or were you able to?
Oh, yes. Yes, okay, so the question is the aquaglyceroporins, so that's a good question. So I studied Aquaporin 9. It transports a little bit of water, but also glycerol and urea. And so it's not, glycerol isn't one of those molecules like water where you kind of think, oh, it's not always obvious the role of glycerol in the body. But it's kind of interesting. This is our hypothesis, and it is held up by studies in mice, but it's not been verified in humans. But the idea is that when you are hungry and you need to break down fat, you break it down into a fatty acid and then also to glycerol. Those are the two components of fat that get released from fat cells. And so the idea is that Aquaporin 7 is in the fat cell, and it lets the glycerol leave the fat cell and enter the blood. And then the liver that expresses Aquaporin 9 wants that glycerol so that it can convert it to glucose. And so the thought is that Aquaporin 9 will be letting the glycerol into the liver cell so that they can convert it to glucose. And kind of the reason why we came up with this hypothesis is that we saw that in rats, if we fast them, then they quickly, dramatically increase the amount of Aquaporin 9 in the membrane of the liver. And I believe it's the part of the membrane, that's facing the blood. And so that's sort of our idea of the role of aquaglyceroporins, at least in the handling of fat and the conversion of glycerol to glucose. So that's one example of aquaglyceroporins and how they might function. So it's completely different compared to water. And then another question that popped up is just what is the most common aquaporin? And I think that one would actually, my guess would be Aquaporin 1, which was the first to discovered because it's present in red blood cells and in the cells that line blood vessels. So those are obviously everywhere. And then it's present in the kidney, in bile ducts, in many, many different places. So that would be my best guess of what the most common aquaporin is, at least in humans. And then another question that's popped up is, has research affected your life? So research has definitely affected my life, just like it's affected everyone and all the things that it gives us in terms of medicine and technology. But it's really interesting to see how getting an advanced degree affects just how you think. So a PhD really teaches you how to think and how to use logic and think critically. And so that just affects the way I see everything. So I think I drive my husband nuts sometimes when he says, oh, well, maybe this is what's happening or this is what's happening. And I say, no, that doesn't make sense because of this or this. I analyze everything. And you just can't stop doing it once you have been trained the way that you are trained in graduate school.
I
I actually had another question, if you wouldn't mind.
That'd be wonderful.
You talked about Aquaporin 9 and the glycerol and everything. So I was just wondering if there is like a mutation or malfunction with Aquaporin 9, could that lead to diabetes, or what can come?
Right, that's a good question. That's a good question, and there have been some studies in mice, but I don't think it's known. It would be more probably likely if it did anything, then it would be that when you were hungry and you were fasted, that your blood glucose could be a little lower than it should be. And so it would, if anything, lead to problems with hypoglycemia when you were obviously hungry. That would be my best guess, but no one has looked at that or studied that.
And I also read somewhere too that there could be a correlation. If we were looking at cancers here, cancerous tumors, is there a correlation between tumor size and the amount of aquaporins present in that tumor?
Right. There's some studies that suggest that there could be a role for aquaporins in certain processes that are important in tumors. So in angiogenesis, the growth of blood vessels, so for a tumor to get a certain size, it needs to have a certain blood available to it, and so tumors often have to grow new blood vessels, and since Aquaporin 1 is in the cells that line blood vessels, then there's been some experiments suggesting that may affect angiogenesis. There's been some studies of aquaporins being involved possibly in cell migration, which is also a process where that is important, for instance, in the spreading of cancer. So I think more studies have to be done, but that's certainly a possibility that aquaporins play a role in those sorts of processes.
Thank you.
Yeah.
Does any other school or anyone have a question to ask of Dr. Carbrey? I would like to ask personally the question of ask what's your family, what do you do for fun, those things. So, put a person behind the scientist.
Right. Yeah, I love to travel. So we do that whenever we can, and I'm also really into backpacking, and my son's in Boy Scouts, so often I'm the only female on a trip with a bunch of Boy Scouts and dads. So those are the things, and I love dogs, so I have several dogs too. So yeah, it's I have a great career that is flexible, and so I would encourage all of you, best of luck to all of you in just pursuing your dreams and going for it, because life is short. So just get out there, challenge yourself, work hard, find what you love, and just go for it.
If we have no other questions, if we can thank Dr. Carbrey, please.
Thank you. You're very welcome. It's my pleasure.
We'll stop the broadcast then now, and I'd like to talk to you off air if possible.
Okay. Yeah.